Hello, my name is Monty Hughes. I'm a professor at Duke University in North Carolina in the United States. And we're here at the International Continents 2024 meeting. And I'm here to discuss a little with two of my colleagues who just gave great presentations in a session in which I was chairing. This is Julien Ratelade and Lysanne Campeau from you said you were with the company.
Yes, my company is called EG427.
Okay, and it's located where?
In Paris, in France.
In Paris,
yes.
Excellent. And you're at the Lady Davis Institute.
Correct, yeah, at McGill University in Montreal.
McGill, okay.
Yeah.
For some reason that wasn't on my notes.
No.
McGill is a fantastic school. We know it well.
Thank you, yeah.
Your presentations were wonderful. Excuse me, Julien, you had some great work with this virus, this non-replicative HSV vector, and it expresses the light chain of Botox. So we do a lot of work, obviously clinically. I don't, because I'm the scientist, but we do a lot of work just injecting it. Do you anticipate that this could replace Botox injections? Is that the idea, to develop it?
So yes, so well, it's not. Yeah, we use this vector, like you said, this HSV1 vector. It's a herpes virus. Mm-hmm. We use this vector because it has very unique properties, very interesting properties. It's a neurotropic vector, so when you inject it locally, it infects primarily the neurons, the sensory neurons, and the wild-type HSV1 goes into that neurons and enters into latency. So the genome is there in the nucleus. It's latent, it's repressed, except one specific region called the LAT region, which is expressed, and we can insert transgenes into this specific region to obtain a long-term expression in sensory neurons. So really use this as a tool to deliver transgenes in sensory neurons. In the case of this study, we inserted the sequence of the light chain of the botulinum toxin. So it's only the light chain.
It's just the light chain?
So it's just the light chain, the catalytic part. So it doesn't diffuse from cell to cell. It stays in the cell in which it's expressed.
Fantastic.
And we block the neurotransmission with this light chain of the botulinum toxin. We believe that with this injection of the vector, so in the detrusor, in the bladder, we can achieve a very long-term effect of the expression of this light chain of the botulinum toxin for years or 10 of years. This is the expectation.
For how long? I'm sorry?
For years or more than 10 years, yes.
So that's a much longer time frame than simple Botox injections. You have to come back often.
Exactly. So we believe that we can, with one single injection, have a very, very long-term treatment. And we also believe that with this treatment, we have much more selective because we target specifically the sensory neurons and the sensory C neurons, because we have a specific promoter driving the expression of the light chain of the botulinum toxin in the C neuron. So we believe we have a very targeted effect on these sensory neurons. And we don't block the contraction of the muscle. We don't block the ability of the bladder muscle to contract. We don't have any effect on the efferent
So there's no
pathways.
effect on efferent.
Yes, exactly. So we hope that this treatment can be long-term effect treatment and more selective treatment.
Fantastic. So if your promoter is the CGRP, correct? Can it activate afferent pathways in other tissues, or is it only because you put it in the bladder?
Yes. So we have a local effect, very local effect, because we inject the vector in the bladder. It's a very big particle, so it doesn't diffuse, it doesn't go anywhere else, and it infects only the fibers present in the tissue. So only the sensory neurons innervating the bladder will express the transgene. And again, the vector is non-replicative, so it doesn't spread from cell to cell. And it's only the light chain of the botulinum toxin, so it stays in the neurons of the bladder. So no, we expect a very local effect.
Local effect.
Yes.
Excellent. So, Lysanne, you were working on brain-derived neurotrophic factor. Is that right? And they had some very interesting work with that. Can you summarize real quickly?
Yeah, of course. So we've had a very strong interest in looking at different neurotrophins and their role in the bladder, and so this came about with a collaborator who makes these different molecules and he's been studying primarily the precursors of neurotrophins and the mature neurotrophins and the impact of these different proteins on different receptors. And so we've had a particular interest in NGF, right? That's something that's been studied for quite a long time in bladder.
Plays a central role.
Exactly. And so back when the study started on NGF, the assays weren't very good at determining how much of the premature NGF and how much of the mature NGF. It would just kind of lump it all together. And now it's becoming more and more obvious that the premature form, the pro-NGF, has a very different role on the bladder and actually leads to more inflammatory reaction.
It's very interesting. Usually you think of a pro-hormone has no activity.
Yeah.
So but in fact, this is more of a pro-inflammatory.
It binds to a different receptor, which is the P75 receptor, while the NGF binds to the more TrkA receptor, which leads to more regenerative. And so, we've been studying that for quite a long time on different models, and because he's developed these different molecules, which one of them is an antagonist of the P75 receptor.
Gotcha.
So it kind of explained the extension of our study looking at BDNF and pro-BDNF, which has, again, a similar aspect of the pro-BDNF has more of an oxidative stress, inflammation, degenerative type of pathway, while the mature BDNF is more neuroregeneration. It acts through similar receptors, and so I think there is definitely a potential role for translation. Some of his molecules of our collaborator actually are now in phase 2 trials.
Oh, and so
we've been looking at how this pro-BDNF, BDNF, pro-NGF, and NGF plays a role in bladder dysfunction. Primarily, we've had an interest on metabolic syndrome of related avoiding dysfunction, so OAB, or and how that could impact or could be beneficial at a treatment level.
So, just to take an interest of my own, we study primarily diabetic bladder dysfunction.
Yeah.
Does it play a role in that? Do you know? Is there
very much so? We actually published. It's not necessarily what I presented today, but we published a couple of years ago in Diabetologia our findings of the P75 antagonist in an animal model of diabetic voiding dysfunction. It was a type 1 diabetic voiding dysfunction, and we found that the antagonist had a significant improvement in diabetic voiding dysfunction.
Oh, really? What,
yeah.
Do you mind if I ask what model you were using?
It was the streptozotocin model.
Okay.
Yeah. But we've been developing different models. We have one model that's hopefully will be published soon, is the Tally-Ho model of diabetic dysfunction.
Yes, I was familiar with some of that. I know one of my colleagues is looking forward to speaking to you today about the Tally-Ho model. We've used primarily the Akita model, which has its advantages and has its disadvantages, as all of these do. We find actually in that model a significant gender difference. Do you find gender differences in your Tally-Ho model?
Very much so. The males develop diabetes while the female become quite obese, but they don't. They become more pre-diabetic than diabetic. Okay. So a very different phenotype.
How about this BDNF work? Is that seem to be gender specific? Is there a role in? Well,
and because we had a prospective cohort of patients, of female patients that had overactive bladder and had different degrees of metabolic syndrome. So we were only interested in female patients because we wanted to kind of remove the BPH factor from that population.
Gotcha.
And so in that population, we published that patients had an increased ratio of BDNF to pro-BDNF, which is interesting. It's actually contrary to what we found for NGF. The NGF story, in this same population, the pro-NGF was higher compared to the NGF. But BDNF was different. So it almost seems as if BDNF is a kind of reaction. So maybe they're trying to kind of regenerate, and so then the mature BDNF increases. But there's definitely also a relationship with MMP9.
Fantastic. Excellent. Well, it's been very nice speaking to you today. You guys did a very good job presenting. I'm looking forward to the progress of your work, and we'll talk more about the Tally-Ho model. And I wish you the best of luck with this vector. It sounds very exciting, and perhaps we can replace Botox injections.
Thank you very much.
Thank you.